It's my pleasure to be here at EBMT and just discuss with you some of the newer options for relapsed refractory multiple myeloma that we've seen emerging in the last 6 months to a year. I think in the relapsed refractory space, unfortunately, our needs remain very, very challenging because patients, obviously beyond treatment with immunomodulatory treatment, proteasome inhibition, and now of course monoclonal antibodies, one of the most challenging areas is now when patients have had all those major classes of drugs, what happens when patients have become what we call triple class refractory. In other words, their therapeutic options have included already immunomodulatory treatments and pomalidomide, bortezomib, carfilzomib, ixazomib, and they've already had daratumumab-based therapy, and unfortunately these treatments have failed them. Where do we stand? So I think in relapsed refractory disease, one of the most important new advances, on the one hand, has been the advent, obviously, of cellular therapies and CAR-T, but in that same immunotherapeutic space has been the excitement around antibody-drug conjugates, next-generation CD38 targeting antibodies. And I say that carefully because isatuximab, which is looking so promising, is truly a chimeric antibody. So really, it's a first-generation antibody, whereas daratumumab is a humanized antibody. So in a sense, isatuximab is actually first-generation and daratumumab second-generation. But in terms of their clinical development, daratumumab has led, and now isatuximab is coming through. And we've seen some very exciting data around the benefit of isatuximab combined with pomalidomide in the phase 2 setting. And we're very excited to see some of the early results from the Icaria trial looking very favorable, and we'll obviously be presenting more of that later this year, and in particular at the ASCO meeting coming up in Chicago in June. Beyond next-generation CD38 antibodies, the antibody-drug conjugate space is really encouraging, I think, with the impact of GSK 916, which the phase 2 data appear very promising around, showing a very high quality of response and a substantial number of the responses, 60%, with just antibody alone in very refractory patients, and most importantly, patients in whom daratumumab's failed them. So that's exciting. Now, beyond the antibodies and the immune therapy space, including, of course, bispecific T-cell engagers, where early data also looks very encouraging, I did want to share with our audience that the small molecule space is looking really interesting. And one of the most promising, in my opinion, is the advent of a completely new class of drugs, the so-called XPO1 protein export inhibitors, or selective inhibitors of nuclear export proteins. These are called SINEs for short, and the point is that the paradigm molecule is a drug called selinexor, and it particularly targets a transporter called XPO1. XPO1 literally shunts out proteins from the nucleus in the cancer cell that would normally shut down the expression of oncogenes and the critical genetic steps that have to happen to allow the tumor cell to resist and survive. If you inhibit that process, these tumor suppressor proteins stay confined to the nucleus and so stop the malignant cell from behaving as malignantly as it does. It's a rather simplified sort of explanation, but I hope it helps. And what we do with this particular small molecule, which is administered orally, typically on either a twice a week or once a week schedule, particularly in combination, is we've seen, certainly as a single agent, that it's been very active in combination with low-dose dexamethasone, even in patients who are what we call triple-class refractory and penta-refractory. In other words, they've been exposed to everything that we reasonably have in the therapeutic armamentarium. And we've seen that about a third of patients respond. We've seen that these responses actually in the responding patients are quite durable. And whilst the side effect profile is challenging, it is manageable. And I think the real excitement around selinexor is going to be in combination, which is literally already well underway. So that's one. Two, there's a very exciting new targeted cytotoxic called melflufen, which is sort of in a different way targeting the nucleus, but doing so in a highly selective way. And this particular chemotherapeutic, but that is aminopeptidase activated and so very specifically targeted, is showing activity in the same sort of population where all the major other classes of drugs have failed the patient, and in those patients we're seeing a clinical response that appears to be durable. So melflufen also of interest, particularly not just as a single agent but also in combination. And then finally in the relapsed refractory space, I think we've been very excited by the clinical benefits seen from the addition of venetoclax to proteasome inhibitors, be they bortezomib or carfilzomib. Unfortunately, venetoclax is associated with a safety signal in association, it seems, primarily with infection. And so as a result of that, there's been a partial clinical hold placed on the development of the drug in multiple myeloma until we've got a better handle on this particular challenge. I would say it's not unexpected because, after all, in the chronic lymphocytic leukemia population, infection from venetoclax was recognized early in its development, and it certainly shouldn't preclude its use going forward, in my opinion, because I think there are ways we can circumnavigate infection and prevent it from being an issue. So I'm very hopeful we'll find our way through this. Having said that, venetoclax clearly is a very active drug, and the results from various studies have shown incredible synergy in patients with translocation 11-14, and even in patients who don't have that sort of cytogenetic abnormality in their disease, we've seen clinical benefit. So I'm very hopeful that once this is sort of figured out and appropriate prophylactic steps are incorporated, venetoclax's continued development will get back underway.